Now let's move on to the investigations, the practical side of this chapter. Here, we have specification number 6, which asks us to investigate the need for chlorophyll, light, and carbon dioxide for photosynthesis, using appropriate controls. So, we'll examine each of these factors one by one and explore how they affect the process of photosynthesis. Before we get into the practical experiments, it's important to remember that safety in the lab is absolutely essential. No matter how simple the activity may seem, safety measures should never be ignored. They help protect you and everyone around you from accidents or harm. For these particular investigations, there are a few key precautions you must follow. First, always wear proper eye protection to shield your eyes from any chemicals or unexpected splashes. If you're using a Bunsen burner during any part of the experiment, handle it with care and be mindful of open flames. You should also follow your teacher's instructions closely at every step. They'll guide you through the process and ensure everything is done safely and correctly. And finally, be especially careful when using stains like iodine solution or methylene blue. These chemicals can easily stain your skin or clothing, so handle them with caution. Once you've taken all the necessary safety steps, you'll be ready to begin the practical work. Let's get started. Let's begin our investigation by exploring the need for chlorophyll in photosynthesis. This is our first practical, and here we'll follow a step-by-step method to carry out the test. This setup and process are designed to show whether chlorophyll is necessary for photosynthesis to occur. In step 1, we start by boiling the leaf in water. This is done to kill the cells and break down the cell membranes, making it easier for the chemicals to enter. In step 2, we transfer the leaf into ethanol and boil it again, usually using a water bath. The purpose here is to remove the chlorophyll. In step 3, check it out. As the chlorophyll dissolves, the ethanol turns green, and the leaf becomes pale or translucent. Next, in step 4, we briefly dip the leaf back into hot water. This softens the leaf, as it becomes quite brittle after being in ethanol. Moving to step 5, we add iodine solution to the leaf, usually placing it in a dish and gently squeezing the iodine around the center. Remember, iodine is used to test for the presence of starch, a key product of photosynthesis. In the final step, step 6, we observe the color changes. The areas of the leaf that were green, meaning they contained chlorophyll, will turn blue-black if starch is present. This shows that photosynthesis occurred there. The white areas, where there was no chlorophyll, remain orange-brown, indicating no starch was made and photosynthesis did not take place. This simple yet effective test clearly shows that chlorophyll is essential for photosynthesis. So, the procedure for investigating the need for chlorophyll is quite straightforward and helps us clearly see which parts of the leaf carry out photosynthesis. First, we begin by placing the leaf in boiling water. This step kills the cells and helps break down the cell membranes, making it easier for the chemicals to work on the leaf. Next, we transfer the leaf into a boiling tube containing hot ethanol. This is done for about 5 to 10 minutes and is meant to remove the chlorophyll from the leaf. Since ethanol is highly flammable, it's important to switch off the Bunsen burner before this step. Instead of direct heating, we usually place the boiling tube in a hot water bath to safely heat the ethanol. Once the ethanol turns green, which shows that chlorophyll has been removed, we take the leaf out and dip it quickly back into hot water. This softens the leaf, as it tends to become brittle after soaking in ethanol. After that, we lay the leaf flat on a white tile and cover it completely with iodine solution. This step is crucial because iodine acts as a test for starch. Finally, we examine the leaf for any color changes. Areas that turn blue-black show the presence of starch, meaning photosynthesis has occurred there. If any part of the leaf stays orange-brown, it means no starch is present, and therefore, photosynthesis did not take place in that part, likely due to the absence of chlorophyll. Now let's talk about what we observe in this experiment and what the results tell us. When we test a fully green leaf with iodine, the whole leaf turns blue-black. This shows that starch has been produced throughout the leaf, which means photosynthesis occurred in all areas. Since the leaf is entirely green, it contains chlorophyll everywhere, and that's why starch is made in every part. In the case of a variegated leaf, one that has both green and white areas, the result is different. After adding iodine, only the green parts turn blue-black. The white areas stay orange-brown. This tells us something important: starch is only produced in the green areas, where chlorophyll is present. The white areas can't carry out photosynthesis because they don't have chlorophyll, so no starch is made there. This simple test helps us clearly see that chlorophyll is essential for photosynthesis. So, to conclude this investigation, the experiment clearly shows that chlorophyll is essential for photosynthesis. Only the green areas of the leaf, which contain chlorophyll, are able to carry out photosynthesis and produce starch. The white areas, which lack chlorophyll, do not show any starch production. This proves that without chlorophyll, the plant cannot make food through photosynthesis.